real world scenes. And we find that even in natural videos, the same GNN architecture can generalize to match human judgments much better than the non-graph model. So it seems like, contrary to what's been found previously, as long as you add the right inductive biases, which I think in the case of social interactions is the fact that they are inherently relational, you can match social judgments with purely visual models. And I think this really suggests that relational information may underlie our abilities to recognize social interactions. So just to summarize, we found that social interactions are selectively processed in the human brain, in the human STS, separately from all sorts of other visual and social information like faces, voices, theory of mind. And that we think relational and visual information in particular may underlie human social interaction representations. And these visual computations are what allow us to so quickly and effortlessly recognize social interactions. So you can go to our lab website or follow me on Twitter to get more updates about this work. And I am happy to take any questions.
Thank you, Leila, for the nice talk. So is there any question from the in-person audience? I guess we have one like a minute for a question.
Hi, Leila. Do you think that the social interaction, I think, is also an area that you correct me if I'm wrong, that's also important for biological motion? Is there a way to separate out those two things?
Yeah. Yes. So good question. It seems like in both our controlled study and the movie study, they are very close by, but separable from each other. So in the point light stimuli, for example, these two, both the interacting ones and the pair of independent agents have roughly equal amounts of biological motion. And while it's very difficult to measure biological motion in a natural video, most of the motion energy in a model is actually due to people moving around. So we think that that was a pretty good control for that as well. But you're right that biological motion is also processed very nearby in the PSTS and also an important social function. And so I think it is interesting to think about how these two types of information interact with each other.
Yeah, that's interesting because they're probably very close together. And yeah.
Yeah.
Cool. Thanks.
Thanks.
Nice. So yeah, if you have more questions for Professor Isik, you'll have more chance during the panel discussion. So thank you for the talk. On to the next talk by Professor Baylor from UC Santa Barbara. Mike, yeah, you can go ahead.
Awesome. Thank you to the organizers for having me. Very excited to be here. And thank you to all of you for sticking around. I'd like to switch gears a little bit and tell you about an exciting application at the intersection of visual neuroscience and computer vision. This is one example where, as a computer vision researcher, you definitely need to know about the neuroscience behind it in order to make a meaningful contribution. And the application I'm talking about is the potential to restore vision to people who are blind with a neuroprosthetic device. Now, as you might be aware, there are millions of people worldwide that live with irreversible vision loss. There are many different causes that cause vision loss. Two of the most common players are retinitis pigmentosa, where you slowly lose your vision from the periphery inwards, leading to tunnel vision, or age-related macular degeneration, where you lose vision in the fovea and the macula first, and then it spreads outwards. Although there are no cures for these diseases, researchers have been trying to come up with ways to treat them. Some of the current approaches that are being studied are gene therapy. And because these diseases are caused by a faulty gene in the retina, the idea is maybe we can replace the faulty gene with a functioning one and thus reverse or at least slow down the disease. A second approach is to use optogenetics to make neurons responsive to light that have never responded to light before. And that allows you to shine a laser on them and activate them precisely. And the third approach, which is what I want to talk to you about today, is the idea of implanting a microelectrode array, either directly into the eye or into visual cortex, to stimulate the surviving neurons with electrical current. Now, this is no longer science fiction. The most famous device, perhaps, is the Argus 2, developed by a company called Second Sight. This device is sometimes also referred to as the bionic eye. It comes with a pair of glasses and a small embedded camera that records the scene. And then frame by frame, the visual scene has to be translated into a series of electrical pulses that are sent wirelessly here with this coil to a receiving coil implanted in the eye that then stimulates the microelectrode array. We can zoom in on this a little bit more to see here the different layers of the retina, and then the implant sitting right on top of the ganglion cells. This particular implant is equipped with a 6x10 electrode grid, each 200 microns in diameter, roughly. So that makes each electrode orders of magnitude larger than an individual cell, something to keep in mind. This device has been implanted in 300 to 400 people worldwide. It is currently discontinued, but there are over 20 new prostheses currently in development. So the field is expected to only grow over time. And even though there are all these different devices in development, some of which will be implanted in the eye and others directly in visual cortex, they all share some common components. And very quickly, they all have an external camera, usually, which is used to acquire visual input. This signal is then sent to a vision processing unit, so usually a mobile device that can do some image processing and stimulus optimization. And then that is sent to the microelectrode array, typically wirelessly through transmitting and receiving coil. Then what happens if we stimulate this device? We will electrically activate neurons in the brain, which is how they usually communicate. So if we do this smartly, the brain might not be able to tell the difference between an artificially stimulated neuron and a naturally stimulated neuron. At least that is the idea. So stimulation of electrodes will produce some neural activity that is then interpreted by the brain as a visual percept. Now, many years of research have focused on this pipeline here. So how do you go from an applied stimulus to the predicted response, either neural activity response or a perceptual response? And what people have done is kind of boiled down the visual processing of the brain into what we call a forward model, basically a black box model that maps stimuli to responses. Perhaps the simplest forward model is just a linear model. We call it the scoreboard model, because it assumes, like maybe you've seen scoreboards in sports stadiums, it assumes that every electrode is kind of an independent light source. And so if you stimulate a grid of electrodes, that should lead to the percept of a grid of luminous dots. If you only have 10 by 10 electrodes, then the produced image might not look very interesting. But if we increase the number of electrodes, eventually we should be able to produce useful vision. Now, this has been used in combination with some computer vision techniques, because obviously what you need to do now is you need to encode a complex visual scene such as this with a low resolution display. And so many different ideas have been explored, such as perhaps we can end-to-end optimize the encoding. If we have a target image, we can learn how to encode that with these small phosphenes, as we call them. Perhaps you want to do it like this, or you want to just predict the edges. Others have thought about, no, maybe what is really important is the ground plane. If you're a blind person, you have to find your way around town. It's important to know where you can walk and where you cannot. So maybe you want to segment the ground plane first and then visualize that. Or maybe you want to combine segmentation with structural edges, which can give you an idea of the room layout, and then combine that into a phosphenic representation. Or maybe it is more important to just highlight nearby obstacles, because that will eventually allow you to react to your immediate surroundings. So many different computer vision techniques have been explored, but all of these suffer from one crucial limitation, which is that this way of producing these phosphenes is not biologically realistic, meaning that these phosphenes cannot explain some of the distortions that have been observed clinically. We know this because we have tried it out. We have worked with real patients and asked them to draw on a touchscreen what they see when we turn on a single electrode. This particular person here, when we turn on one of their electrodes, they would draw a line. And that seemed very odd at first, so we repeated it. And we see that over many trials, the person would always draw the same line very consistently. So what we can do is we can align all these images by their center of mass and average them to get a good idea of what this particular electrode, here called D3, would produce. Here's another example. These are all vertical lines aligned by the center of mass and averaged. You get a pretty good idea that this particular electrode always produces a vertical streak. Repeating this for all possible electrodes in the array, we find that all of them produce different streaks, but they're repeatable across trials, right? Because this is averaged. You see that over time, the percept is repeatable. It's just that going from one electrode to another, the orientation of the line changes drastically and seemingly randomly. This is just one patient. So repeating this experiment for different patients, we find a whole repertoire of different phosphene shapes. They include lines and triangles, wedges, all sorts of things, but most of them I would not characterize as a pixel or a blob. And so where do these distortions come from is a big open question in the field. We could either spend 10 years doing psychophysics to have a data set that's large enough for a deep net to fit, or we could dig in a little deeper into the neuroscience and see if we can find underlying causes of these distortions, as we call them. So to give you a really quick crash course of the retinal physiology, you might already be aware that when light enters the eye here, it gets refracted and projected onto the back of the eye, which contains the retina. And the retina already is a maybe 3 or 4 layer network that processes the incoming visual signal. We have our photoreceptors, the light sensitive cells, that then send their signal to bipolar cells and finally to ganglion cells, which are the output layer of the retina. And they send their signal through these axons that go to the optic nerve to the back of the head where the visual cortex sits. Now, if you have one of these blinding diseases, such as retinitis pigmentosa, what happens is you lose your photoreceptors. They start to degenerate. And so the idea here is to replace the lost functionality with a chip. If you put it right there, it's called subretinal, but there are other locations where you could put your device. The one I just showed you, the Argus 2, that's called an epiretinal device. And you can see it sits here on top of the ganglion cells. But not just the ganglion cell bodies, it also sits on top of the ganglion cell axons. And that turns out to be the key for these phosphenes. Here is now a bird's eye view of the back of the eye. And every one of these lines is a fiber bundle, an axon that has been extracted from 55 cadaver human eyes and aligned by the optic disc and the fovea. And what you see is that these fibers form a very stereotypical pattern. So now we can, I guess, ignore the math. We can explain this with a spiral with a very small number of parameters. And we can start to ask what would happen if we had an electrode right there and we stimulated that neural tissue. What would happen is we wouldn't just stimulate the ganglion cell bodies that live directly below the electrode. We would also stimulate the ganglion cell bodies that send their axons through this point in space to the optic disc. And all of these happen to fall on a line, because if you're a neuron that lives over here, you will send your axon along this pathway. And so this one, same for this one. So by activating them all at the same time, we're telling the brain that we saw something and it was right here, but it was also right next to it and right next to it and right next to it. So essentially what we're doing is we're producing a line, a streak. There's another structure, landmark in the retina that is important. That's the horizontal raphe. That's a region where there are no axons. The reason for that is if you are a neuron that lives directly above it, you will send your axons here, up here, and over to the optic disc. And if you live right below it, you will send it downwards. Okay, so skipping many years of research, we were able to come up with a computational model that can predict what a phosphine will look like depending on where it sits on this axon map. A schematic is shown right here. If you now stimulate a certain electrode, it is assumed that the current will spread not just radially, it will also spread along these axon bundles. And we can parameterize the spread with two parameters, rho and lambda. So if you make rho larger, if you make rho larger, you should get a blobbier percept. And if you make lambda larger, you get more of a streak. But that's not all. The shape of phosphines also depends on the stimulus parameters. For example, if you increase the amplitude, phosphines tend to become more blobby. And if you increase the frequency, the shape doesn't change, but the brightness does. And then finally, if you change the pulse duration, you can get rid of some of the streakiness and turn the phosphine more blobby again. So all of these are nonlinearities that are not taken into account by the traditional scoreboard model that a lot of people tend to use. And therefore, predictions with your scoreboard model might be highly misleading. To help people understand the quality of these phosphines, we have developed software called Pulse to Percept, which is a Python package you can all download and try out. And it can predict for all these common retinal implants that are out there, what the vision generated by them would look like. I wanted to show you a couple examples here for fun. So here we're simulating what an Argus 2 might produce. Remember 6x10 electrodes? If you were in the subway and you see people walking by, without this axonal activation, you basically would just get a downscaled version, grayscale version of your input with a 6x10 grid. But as soon as you add the axonal activation, things start to kind of smear out and get elongated, which you can appreciate most here. Again, maybe 6x10, that's just not good enough. So what happens if we increase the number? For reference here, again, 6x10. What if we now had a 12x20? And you can start to make out more details of your immediate surroundings, but it's also evident that you get kind of this fisheye effect, right? This local distortion and bending of the individual pixels, if you will. So there's an opportunity here to optimize the stimulation, not just go pixel by pixel, but incorporate knowledge about the distortions to produce better stimuli. And so going back to the schematic I showed in the beginning, here's the setup where you go from stimulus to response. What we really want is not this forward model that tells us for a given stimulus, what is the response. We want to know the opposite. We want the arrows to flow in the opposite way and tell us, how do I achieve a target response? And so one way to solve this, or to think of this, is in an autoencoder structure, where your forward model is fixed because it's informed by neuroanatomy, behavioral results, all sorts of things. And what you want to learn is an encoder that inverts this forward model such that some reconstruction error is minimized. To make it clear in the application of a visual prosthesis, you would start with a target percept, it could be an image or a movie, whatever. And you would feed it through an encoder that you now want to train such that the stimulus it predicts, if you feed that through your forward model that is fixed, it should produce a percept that looks just like your target percept. We call this a hybrid neural autoencoder because the forward model is typically a symbolic or a numerical model informed by the neuroscience. And the encoder is just a deep net that is trained to invert this fixed model. Often it's impossible to invert this fixed model because it's either too computationally expensive or it is not differentiable. So some people instead use a surrogate approach where they train a deep net to approximate the forward model and then use that in an end-to-end fashion. So we wanted to see how both of these approaches work, one where you have the fixed decoder in the loop, and one where you use a surrogate model, and compare it also to the naive approach. As a reminder, naive approach just means whatever image you have, you basically paint it on the retina. And then depending on your rho and lambda, the output will look better or worse. For example, if rho and lambda are really small, then yes, you basically have this pixelated model that a lot of people assume we have. But we also know some patients, they have much larger lambda, so much streakier percepts. And if you just apply the number 3 there, then you get something like this, and it no longer looks like a 3. And the same is true if you increase your rho. So different patients have different values of rho and lambda, meaning that this naive approach only works in the most ideal cases. However, the hybrid neural autoencoder seems to perform quite well in a range of these different rho and lambda values. Here tested on the MNIST data set, because everyone loves MNIST now. So here we have some target images and some results for small values of rho and lambda. You can see the HNA is performing really well, producing a crisp image that fits the target very well. But the naive is not doing so bad either, as pointed out before, because rho and lambda are small. Interestingly, the surrogate model, where we trained a deep net to approximate the forward model, is already performing badly. And we will see that gets even worse as we increase, for example, lambda here. So now in the naive model, you should see long streaks. You also see it in the surrogate. But if trained end-to-end, the HNA, the hybrid model, can learn to get rid of these streaks that you would get from a naive encoder. It learns to compensate for them. And so the same is true as you increase rho in the bottom row, Greek rho in the bottom row, and lambda as well. So you can see in the naive encoding here, you just get a blob of light basically, whereas the hybrid encoder can still perform pretty well. To show you that this also works somewhat on more complicated data set, we also tried on MS Coco. And here to help the network a little bit, we segmented the main object in the scene as shown here. So this is the target image. And then you can see the prediction of the hybrid model and the bottom row, the naive prediction. And so while the hybrid models is not perfect, there's a lot of detail is lost, we did some additional studies to show that this is mainly a limitation of the number of electrodes. Here we used 15 by 15 electrodes. And if you increase the number with the same model, you actually get a much crisper, much better result. So this seems to be a very promising strategy in general. But I personally believe that this doesn't go far enough. To tell you a little bit more about how these devices are typically used is that each company comes up with their own stimulus encoding, how to go from video to stimulation, electrical stimulation. Often this is proprietary because they might use some fancy computer vision image processing. But then there's this one encoding that is being validated on different external tasks. So basically there's a hope that this provided encoding, this provided vision will be good enough to support tasks such as visual acuity, object recognition, or navigation. And instead what happens is that people say this artificial vision is very different from natural vision. And often it's not good enough for user expectations because they have to spend weeks and months of training to be able to use this new visual language. So instead, what we're proposing is that the optimal encoding might be highly context dependent, right? So if you are interested in visual orientation, like finding identifying objects in relation to you, then you might rely on one encoding, and that might look very different from if you're doing a mobility task and you're just trying to get from A to B. And it might be even more different if you're just interacting with people. There are different visual augmentations that could help here, right? Some might include object segmentation, others might include highlighting nearby obstacles or faces. But the bottom line is that there should be different modes that focus on functionality. So rather than making any claim about restoring vision, we should provide a tool set where the user can choose the appropriate task, the appropriate encoding for whichever task they're engaging in. And so this would move away from clinical tests and focus more on the real world tasks and find the visual augmentation strategies that optimize performance on specific everyday tasks. And so we call this the smart bionic eye, because it would provide AI-powered visual augmentations to support specific tasks. Again, if you are outdoors and you're navigating, maybe it's just important to know where nearby obstacles are, maybe cars and people, but you also need to kind of have an understanding of where you are in relation to your surroundings. So perhaps there is a computer vision-based pre-processing strategy that would allow a prosthesis user to perform well in this particular task. How do you find the best strategy? That's really hard. So what we propose or are already doing is we're using a virtual reality prototype. Because we have this forward model, we can have a system that shows you the simulated prosthetic vision in a VR headset. And so then we can have sighted users act as virtual patients and perform the tasks we're interested in while we try out different augmentation strategies. In the end, this focuses a lot more on the user and the end user experience. And so it's moving more towards an iterative design process with the end user in the loop, such that you can really find the thing that maybe doesn't look the best, but it provides the most functionality. You're all aware that there are a myriad of strategies that could be useful here, since deep learning can operate on single RGB images to segment objects or to even estimate relative depth, for example. There's methods to find the structural edges of the room layout, and there's things that haven't been explored in this application at all. For example, visual question answering, where you could ask, for example, where did I place my keys again? And while you look around with your bionic eye, it would detect the keys and then outline them for you. So once these computer vision systems get more sophisticated, there are a lot of exciting avenues to pursue. I just want to make the point that it is less about dreaming up these new augmentation strategies, because there's a lot of them. And it's more about really identifying the visual cues that best support behavioral performance. A quick example most of you might be familiar with is optic flow. So we know that in texture-rich environments, we rely on optic flow to navigate, but we're also able to switch to different strategies in less cluttered environments. And for example, navigate more by landmarks. And these abilities change with vision loss. So how can we know which cues, which way to augment the scene is going to be most supportive for these particular end users on a particular task? Then finally, our implementations have to be efficient, because they have to go on the visual processing unit, and they have to be robust, right? Because if you miss an oncoming car, it could be a life or death decision. So it really requires designing systems with predictable failure modes, not just working at the edge, but really being able to allowing us to understand how they make the decisions. So that goes into explainability, but I also understand when and how they fail exactly. And so to wrap it up, I believe that computer vision has the potential of drastically improving the artificial vision generated by a bionic eye. But however, to provide meaningful solutions, we need to understand the neuroscience. And that might involve using these realistic phosphene simulations that are psychophysically validated, such that our predictions can be more or easily translated to the real world use case. And there's an engineering challenge with designing efficient and yet robust systems that have predictable failure modes so that we can use it in the real world. With that, I say thank you, and I'm happy to answer your questions.
Thank you, Michael, for the really interesting talk. Is there any question from the audience? Or from Zoom attendees? I have a question. So you use of this prosthetic device, will it require training of the user? Like, what does an EDS mean, or what does a blob mean? Is it an EDS or is it a deft map? I mean, they may mean different things to different people.
Yes, it will definitely require training. A lot of people describe their new vision as kind of like fireworks, fireworks show. You see these flashes of light that appear, and they seem very unnatural first. And second, require you, therefore, to associate certain forms and shapes with meaning. So people have to be trained usually, both in terms of their strategies, like how to sample the visual scene, how to move your head to get the important information. And then also in terms of interpretation, what do these shapes mean and how can I make use of them?
I see. I think Jeff has a question.
Yeah, the transforms you were looking at were very specific to the placement of the prosthetic, right? Because you were placing it on the bundle. So what would be the optimal place to, with being not too invasive, to place this prosthetic? And how would that change what kind of algorithms you'd have to use?
Yeah, that's a great question. I don't think I have the backup slide for this, but we looked at this. In the retina, you don't have cortical magnification for one, so it's a little easier to place the array a little more parafoveally. But with simulations, we looked at what would be the best orientation and location to place the array, and it turned out to be really close to the raphe and basically flat, because that kind of minimizes your streaks. But the answer would, of course, be very different if you now go in V1, where you have this strong cortical magnification. And so currently, you would have to train a different encoder for every location, for every patient, basically. We're trying to overcome that limitation, but yeah, it's hard.
Thanks.
Mm-hmm.
Yeah, very nice. Yeah, really interesting.
Thank you.
Any other question? If not, then next up is, oh, sorry, we have a break now for half an hour. It's 3:30, so we'll take a break until 4 p.m. And then we have 2 more talks and 1 panel discussion, then the awards and closing ceremony. So yeah, hang on. See you in half an hour.
Hey, Bruno, guess
Okay.
you're up next. How you
Yeah.
doing? Nice to see you.
Hey, good to see you. Yeah.
Hey, Bruno.
Hey, hello, hey, Rock. I was just going to ask maybe if I could just test to make sure I can share screen and everything.
Yeah, please. Yeah, can go ahead.
Okay. So let's see if I do this in full screen mode. Okay, do you see that?
Yes, we can see our full screen. I mean,
Awesome.
yeah.
Okay. Okay.
So yeah, your talk is up in almost half an hour. Yeah.
Yeah. Right. Okay. I'll just hang tight here. Whoops. Oh, stop share. Okay.
So I'll use this opportunity to advertise a upcoming specialist on the same.